Today, I'll be carrying out an experiment to investigate the action of catalase. Before we start, I'd like to tell all of you about catalase and hydrogen peroxide. What is catalase? Catalase is an enzyme present in the cells of many plants and animals. Cells with a high metabolic rate have a high concentration of catalase. This is the following equation that involves catalase. As you all can see in the equation above, it catalyzes the breakdown of hydrogen peroxide to water and oxygen. Now, I'd like to tell all of you about hydrogen peroxide. Hydrogen peroxide, or better known as H2O2, is well known as a cell-damaging agent that is produced during normal cell metabolism of aerobic organisms. For example, when the immune system is activated in response to bacteria, large amounts of H2O2 are produced by certain cells to fight the infection. Why is H2O2 dangerous to our body cells? This is because it is an excessive production of oxygen metabolites, which leads to oxidative stress and diseases. Now, if the level of H2O2 becomes too high, the cell's DNA and other proteins can be damaged. Scientists suspect that this and similar processes are what leads to cancer, diabetes, and many more diseases. Now that I've explained about catalase and hydrogen peroxide, let's get started with our experiment. So here are some materials and apparatuses you need for this experiment. One of them is the hydrogen peroxide, but of course we won't be using all of them. We'll be only using a small amount. A piece of chicken liver, 1 raw, 1 boil. A potato, 1 raw, 1 boil. 4 containers labeled A, B, C, and D. Finally, a stick from a penyapu lidi, which you can use it as a glowing splinter if you use it with a match. Before that, remember to wear your gloves and of course the face mask. Let's get started. Alright, so first I'm going to take all the food over here and I'm going to cut them into half. Now the reason why am I doing this is because we don't want to waste the material, so that's why I'm gonna cut all of the food into half. I'm going to place the food into their respective containers. First, we'll start with the raw chicken liver. The other half we're just gonna put it back here. Alright, fast forwarding from here. Some hydrogen peroxide into the containers. And remember, be very, very careful. Oh! Okay, there is no reaction happening here. Also here. Now we're going to be using a glowing splinter to test the presence of oxygen in each of the containers. Alright, so we'll start with container A. Alright. As you can see, it burns vigorously, so there's actually a lot of presence of oxygen in there. Alright, now container B. Oh, it burns a little bit. We're gonna put it in container C and we're gonna see what happens. Oh, it burned off completely, so this means that there's actually no presence of oxygen in here. And now we're gonna put it in D and we'll see what's gonna happen. And yes, it burned off completely again. And just like say, there is actually no presence of oxygen in here. After doing the experiment just now we all know that hydrogen peroxide reacts to catalase, which are present in both the raw chicken liver and the potato, causing an oxygen from H2O2 to split off, creating the bubbles we see. As you'll noticed, the cooked chicken liver and potato produced from little to no bubbles. And this is because the heat degraded the catalase, making it incapable of processing the hydrogen peroxide. So that's all from me today. Thank you so much for watching this video. I hope all of you have learned a thing or two from this experiment, and always remember, biology is study for life.